So our next speaker is, Laila Kaasik. I hope I pronounced this name correctly. And we are going to listen about the sensors for determining single droplet acidities in the Venusian atmosphere. So how to deal with Venus and what can we measure there. Go on, the floor is yours. Can you share your screen? Can you say something so we can check it?
Can you hear me?
Yes, yes, I can hear you.
Great.
Perfect. Awesome. And
yep.
Awesome. Perfect.
You can see it, right?
I can see it, and I will let you know when 10 minutes is passed and then 15 minutes.
Yeah, I can't see anyone else anymore, but I hope you can see me and hear me well. Hi.
Very good. Go on.
Okay, good. Hi, I'm Laila Kaasik from Tartu Observatory in University of Tartu in Estonia. I've been working on a sensor for determining single droplet acidities in the atmosphere of Venus. We recently published a paper about it, and I'm going to give you a quick overview what's been happening, what we've been doing, and what is it all about. Probably Venus isn't the most familiar subject in terms of astrobiology to many, so I will start with a short overview of what we've been doing and why it's happening and what's happening on Venus in general. So this whole thing I'm doing, the sensor, is part of a Venus Life Finder project led by MIT. It's a series of missions. More information can be found on their website, Venus Cloud Life, and you can go and check out there. We published recently also a mission study for the whole mission for the other instruments, and our instrument is also mentioned there. In the 60s and 70s, Venus was the destination and first ever successful interplanetary voyage and landing ever. But final local in situ measurements on Venus were conducted in the 80s. So it's been a really, really long time coming, looking into Venus again. And now, luckily, there are multiple missions to Venus planned by other institutions, but also this Venus Life Finder series that's led by MIT, and I'm participating in with my group. So the mission itself consists 3 different missions. A small mission is starting soon, and it's done in collaboration with Rocket Lab. It's a small, tiny mission just to check general conditions there. Then there will be a medium mission, hopefully in late 20s, and that's the mission that our instrument will be hopefully part of. And there is also planned a large mission, which is a sample return mission to Earth from another planet that also hasn't been done before. And there are works going on with that in Mars, so it's interesting and which one we get to first. So introduction, what do we know about Venus? We, as I said before, their last local measurements, in situ measurements, were done in the 80s with outdated, by this time, outdated technology. But what we do know is the surface temperature is very high. It's 462 something Celsius. No life, as we know, is possible on the surface with that pressure. No. Thick clouds, however, it has a thick atmosphere, thick clouds that have a temperate zone. And could the clouds maybe be habitable? We know on Earth there are also organisms found in the atmosphere, in the clouds. Could maybe this even thicker cloud cover have habitability potential? Let's just say that. So these clouds aren't like our clouds. They consist mostly of sulfuric acid. And the main question is how acidic actually is it? Is it the concentrated sulfuric acid like it has been thought? Or are there more different kinds of droplets with different acidities in the atmosphere. So also there are other anomalies and uncertainties like anomalous gases, an UV absorber that creates interesting patterns when you look at the UV light. And there's also a little bit too much molecular oxygen. There's ammonia detected organics, but are these artifacts or are they real? We don't know really. We will keep arguing over it until we actually go and see. So how acidic? The early consensus was that the Venusian clouds are composed of near concentrated sulfuric acid, but there are models that suggest now that the pH range could be between -1 and 1, and this implies some sort of neutralizing agent. These works have and it also means that there's some complex chemical cycles that we don't really know about. So even if there's not life, there's something interesting to look at anyway. Venus, we usually imagine, is a lot of people think of seeing like the image we see on the right here. But actually, when you look at Venus, it's like a hazy ball. Even this image here is taken in ultraviolet light. It has some patterns. You see, some have compared it to like algae patterns that form on Earth. But that's, of course, that's nothing certain. It's just an idea. But there hasn't been found the source of that. So here is also an image of the possible balloon mission that we have planned that will also include our instrument. So going into the instrument and the paper we are been working on and published, this is the Tartu Observatory pH sensor called shortened TOPS. Here, the whole idea is to have a camera that's pointed at the sensor plate. Sensor plate consists of a pigment and a substrate and the matrix to bind the pigment on the sensor plate. The camera will be looking on the sensor plate, and there are exciting LEDs that will excite the pigment, and depending on the acidity of the droplets that fall on this sensor, we will read different spectra from that. So the pigment that was chosen is fluorescein. This work has been mostly done by one of the authors of our paper, Ida Rahu, who's a chemist. And we tried also other types of pigments, but when I joined, but we found that this is the most suitable for our needs. We can see there's clearly a concentrated sulfuric acid. We can see nice blue tubes, and on the other end, water green tubes. So very visible differences, as we can see with any pH sensor, the papers we have used in school. Basically a very similar concept, but we used a little bit more different system here, and it needs to be also working on the very high acidity range, and that's the very important part here. For that, to use that pigment, we need to bind it to some medium. We need it to be on the sensor, and so we tested. When I came in, we tested some different matrices that we could use. The matrices were polyvinyl alcohol, PVA, and naphthalene. There have been other ideas also, but these the ones we tested. We saw that polyvinyl alcohol is very suitable for our needs. It's possible to create very thin films with it and suspend the pigment in it very easily using a bar coater method here, but other methods were also used and tried. Here is a naphthalene that we also used as a commercially available, but that didn't work because it wasn't. It was hydrophobic, so it wouldn't absorb. The droplets wouldn't absorb into the medium. So if we have this sensor plate on a glass. Pigment is there, and we have the matrix. We have to simulate the clouds of Venus to test if it actually would work. Here you can see a video of me spraying some mist there. It was done with a spray, just a simple perfume spray, basically. And the other side, you can see we're using an ultrasonic atomizer. That creates smaller droplets, and it's easier to put into some kind of to automate it, basically. Here are some tape and films to protect it from sulfuric acid, because we're doing this also with concentrated sulfuric acid. Also, we tried it with water before, so the ones where everything is out in the open, it was just water. So a lot of playing around with water and electronics. That's been an interesting experience. But basically, yeah, ultrasonic atomizer, it's a speaker that does the mist thing. So basically, these are like commercially available things, and what we used is quite similar in principle, actually. And droplets under the microscope, the droplets we created, they are visibly different. Concentrated sulfuric acid, as you can see, is a different color than more dilute versions. And also we were able to produce small enough droplets that are actually suitable for our sensor, because we need the droplets to be around the same size as the real droplets in the atmosphere of Venus we're interested in, especially the bigger ones that could potentially be habitable. Here we can see that the size is around the same ballpark as the bigger droplets that we are interested in. But more research into this is needed with better equipment, better cameras, better microscopes. And here is also an example of how different acidities can be seen. Concentrated sulfuric acid, 9 molar, 1 molar added each other, and you can see the differences in different illuminated with different LEDs.
12 minutes.
Yeah, cool. Thanks. And here is some experiment with the water here in this case, because it's out in the lab, but just to see how it lands there. You can see the fluorescence happening. And in the end, we had to, of course, test if it's possible to actually measure something out of it. So this is what I built in the lab, what I had available. We're still working on it. And for this paper, we just managed to put something together that will show that we can actually measure the acidities, the different concentrations based on this idea with this pigment. Here are two exciting LEDs, and these are the graphs that came out of it. There's a lot of noise, there are a lot of human error here involved, because it was done manually by me, a biologist, I haven't programmed it to be automatic yet, but we have a new student coming in to help to automate everything and make it all more exact. And here is, yes, if you're more interested in that, then we have a paper out. The name is here. And also I have this popular science blog with our colleagues, that we have this one short article on about searching life on Venus. That's an easy read probably for anyone in English. And thank you a lot for your interest. And if you have any questions, you can also ask me later on and contact me and add me anywhere.
Perfect. Thank you very much. Fascinating talk, and let's hope that we will very soon get some new data from the from Venus, atmosphere of Venus in this case. And we have two questions for you, and about 5 minutes for you to answer them. So first question from Priya Das Sarma. Could you tell us a little bit more about the microscopy used to visualize the droplets?
Yes, it was quite a cheap microscope that my boss actually bought for soldiering purposes, and we also used that. So it's not possible to. You could see the small droplets there, but we need better equipment and also better camera to attach there. It's a regular binocular, nothing fancy, just what you have in a space tech lab. So that's just to get some first images, but we're definitely looking into getting better equipment for all that.
Perfect, thank you. And the second question from Jan Spatek. First, Laila, nice talk. Second, how do you distinguish between small, highly acidic and large, less acidic droplets, since the sulfuric acid is absorbing into the substrate?
Yes, we have to really study more the aspect of how different droplet sizes react. So we really have to see that, because right now the measurements we did in the end, we did just measure the spectra. We didn't use the camera, but in the end we will use a camera that will see it on impact the size of the droplet also. It will affect, but yes, that's one of the things we need to look more into. Right now my team thinks that there shouldn't be a big difference, and we haven't seen a large difference depending on that, but because the main thing is that it's dry. The PVA is completely dried, and the water has been removed from it. That's one of the main goals, that to get a really dry PVA, so it wouldn't be diluted in water in that case. But if it's overlapping with other droplets, then that's basically that sensor lifetime is done for that screen at least.
Perfect, thank you. Next question from Matthew Marsky. How viscous is the Venus atmosphere in the temperate zone of the clouds, I guess? Would prokaryote-sized cells be able to swim in it?
Sorry, yeah, the droplets in the clouds, and if the small organisms could live there. Yes, I also did my master's thesis on it and discussed it with a lot of biologists and had to defend it in front of biologists. And nobody thought it would be impossible, because the sizes of the bigger droplets are big enough to have small organisms in there. Like prokaryotic organisms could potentially live inside these droplets. There also could be a life cycle where they drop out and dry out, and that's explained in a paper by also from the MIT group who is heading it. So if you want more information about the possible organisms that could potentially live like this, I can send some links.
Perfect. And another question from Priya Das Sarma. Do you expect to find particulate matter in the atmosphere along with the droplets? And I guess how would you imagine this could influence your sensor measurements?
Yes, actually, we've been thinking about including, right now we're just doing different concentrations of sulfuric acid. Of course, it's not exactly appropriate for it. So we've been thinking about putting in organics, putting in other particulates in there to see how it actually affects it. But that's next stage. We're actually building up our new lab now, and that will be the next stage, trying to see that how it affects the sensor.
Perfect. Very, very interesting. And if you have any more questions for Laila, I will suggest you contact directly the speaker. Yeah. And right now, perfect. I guess you will hang out for the next session as well, and the next talk and the next session.